Hello and welcome to Nikolai's genetics lessons, and today I have prepared two easy questions for you. And the first question is: in bacterial cells, ribosomes are packed into the cytoplasmic matrix and also loosely attached to the plasma membrane. What is the function of ribosomes? I know that many of you know the answer, so I want to give you some extra information about ribosomes, and I want to clarify some confusion. So imagine that this is messenger RNA, and ribosomes consist of two subunits: small ribosomal subunits and large ribosomal subunit. And in prokaryotes we know them as this ribosome as 70S ribosome, and in eukaryotes as 80S ribosome. And also some of you know that small ribosomal subunit in prokaryotes we call 30S ribosomal subunit, and large one 50S ribosomal subunit. And in eukaryotes, large one 60S ribosomal subunit and small one 40S ribosomal subunit. And in prokaryotes, as you see, 50 plus 30 would be 80 and not 70 as we see here. And in eukaryotes, 40 plus 60 would be 100, but it's strange that we have 80 as here. So what these numbers stand for? Some of the students think that these numbers stand for the mass of the subunits, but this is not actually so. These numbers stand for the movement of these subunits when we centrifuge them. Speed of movement, and on the speed, what factors would influence? First of all, would be a weight. Second factor would be shape of the subunit, and third would be density. So that's why we have numbers here that doesn't add up to be, for example, 80S, but instead we have 70S here, because as ribosome as one unit would have different speed than two subunits separately centrifuge. And S here stands for the Svedberg coefficient, so actually this is not a weight as many may think. So and also I hope many of you, or all of you, know that ribosome would be a site of protein synthesis, of course with the help of the tRNAs that bring specific amino acids that's specified by codons that we can find here on the messenger RNA, and tRNAs would have anticodons. So as you see, the correct answer would be answer B. Ribosomes are sites of protein synthesis. Next question: plasmids are important to the genetics of many bacteria. This is because answer A, they inherited from one generation to the next. This is true. Bacteria would be propagated by binary fission, and also mother cell would or daughter cells would share 50% of the cytoplasm of the mother cell, and also would share plasmids that can be found in cytoplasm in many copies. And answer B, they may carry genes that give their host a selective advantage. This is also true answer. And answer C, they can render bacteria drug resistant. And because we said that answer B or statement B is correct, so plasmids may carry genes. So some of these genes also can provide drug resistance to bacteria. So as you see, the correct answer would be answer D, all of the above. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. share this video with your classmates, and see you in the next video. Goodbye.